Let's see. First of all, I want to thank the organizer for inviting me. I think I'm the only theory talk at this symposium. I'll speak about some work which is so far unpublished, preliminary, quite possibly completely wrong, but looks very nice. I'm quite excited about it. Main criticism is that it's probably too nice for biology to be true. So it's theory, so it's top-down. It's about the ventral stream, so it will apply to the mouse insofar the mouse has a ventral stream, which probably has in some primitive way. Let's see. Hopefully, I'll get some slides first. Should I plug and unplug? Let me first give you some quick background about a model that we have been working on for the last 10 years or so in my group, which is trying to be a summary of a lot which physiology, thank you, and anatomy has learned about the ventral stream. This is the ventral stream in the macaque monkey, this series of areas from V1 to the infratemporal complex, which is believed to underlie at least some of the computations involved in object recognition, in particular in answering the question, what is in the image? The size of the areas there reflects the number of neurons in each one of those areas for a total of about, I would say, 700 million or so in the monkey brain. So the basic story that comes from several decades of physiology is that as you go from V1 to IT, the features to which individual neurons respond become more complex, going from oriented contours in V1, simple and complex cells, to V2, to V4, and posterior and anterior IT. This is a cartoon, not to be taken too seriously, but depicting this increasing complexity of the visual features that activate these neurons. And in parallel, you get more and more invariants, starting with complex cells in V1, which, for instance, according to the description of Huber and Wiesel, pull inputs from simple cells in slightly different positions and therefore keep the same selectivity but have some invariance to position, as it turns out, to scale. And as you go up to anterior IT, you get larger and larger receptive field sizes and more invariance to position and scale. OK, and so this model we have been developing has several layers reflecting different areas of the ventral stream, starting with simple cells, these S1 layers. Simple cells of the same selectivity are pulled together in complex cells that therefore are more invariant to position. The simple cells themselves are doing essentially a dot product, a kind of template matching operation through their synapses with the input patch of image that activates them. And as you go higher and higher, you get more complex features. For instance, in S2, there will be more similar, again, simplifying a lot, to combinations of lines, say corners or so, and so on as you go higher up. And at each level, you get, therefore, at the S level, you get more specific features, more complex features, and more invariance at the C level. This is just probably the last one, maybe the most detailed one, of a large class of models. The first one being in words what Hubel and Wiesel said long ago, and then Fukushima was the first quantitative model. And feed forward, we have been trying to be quite quantitative and so on. And we managed to reproduce a lot of the properties of cells in different areas, predict some of those properties. I'll not show you the whole list. What really surprised me more is the fact that these models, which were derived from properties of cells and physiology of the monkey ventral stream, actually performed as well as computer vision, the best computer vision systems on computer vision tasks like labeling, classifying, about 10 different type of objects in databases like this, like cars and trees and buildings and so on. Or in tasks like this, where essentially the same architectures is used to classify micro behaviors in a mouse in a cage so that the behavior can be quantified automatically with a performance that is at the level of human technicians. So the surprise was that it works pretty well at the computational level. And it has been 10 years. Until 3 months ago, I did not quite understand why. So that's actually an interesting irony. We had the quantitative models. We could do simulation, experiments with it, ablation, lesions, whatever you want, mutations. And I did not get a good intuition of what made it work, what it was actually doing, whether 3 layers were better than 5, and under which conditions, and so on. So that's why we need a theory. And now I think I have one, as I said, could be wrong biologically. And it's kind of ambitious. It says, it assumes, you make one assumption. It's about what the ventral stream is actually doing or computing. And from this, it tries to predict the hierarchical architecture of cortex, the properties of cells in visual areas, the existence of high-level modules like face modules. OK, so the first point is what is the goal, the computational goal of the ventral stream. That's the assumption. It starts with this question. If we have tasks of identification, I have to recognize, I've seen, say, once Christoph's face, and I want to recognize it again. Identification. So clearly here, the difficulty of recognition is the fact that the second image could be under a different distance or illumination or viewpoint. So invariance to viewpoint is the key part, the key difficulty in identification. For categorization, my claim is that it's probably the same. The difficulty is due to viewpoint. Here is just a motivating example. Suppose I give you one example of a dog, one example of a horse, and then this is the training, and then I ask you, showing a new image, whether it's a dog or a horse, and the question is the difficulty of this categorization task more because there is intra-class variability, there are many different dogs and horses, so one exemplar cannot be enough to learn the class, or is more due to the fact that I can show you images from different viewpoints, distances, illumination. The claim is that it's more the second one, much more. And as a motivation, we have normalized these images of dogs and horses so that they have the same viewpoint as here. And if you train on very few examples, 1, 2, 3, of horses and dogs, a very simple classifiers, even just nearest neighbors of pixels, it does pretty well, a very small number of examples. So if the ventral stream could somehow factor out the viewpoint, then the rest should be easy. That's the claim. Okay, so the claim is that the architecture of the ventral stream is producing, let's call it a signature, would be a vector of numbers. It's a simplification, a vector of numbers, I don't know, 20, 100, corresponding to firing of high-level neurons, which is a signature for the image or the object. And you want this signature, you will use this signature as input to a classifier or to an associative memory to retrieve the label or other properties of that object. But you want this signature to be ideally invariant as much as possible to transformations like different viewpoints or different distances or different translation or scale. So this is the summary of what I'm going to tell you. I'm going to tell you how the ventral stream may learn these transformations, mainly affine transformation in 2 dimensions, translations, rotations, scale, and combinations of them, and become invariant to them. And then, and this is by assuming that there is a little neural modules, basically simple like cells, which just store essentially frames of objects transforming, moving, a small number of them, and by storing them can then later in adulthood perform a very simple computation with new images that makes them invariant to these transformations. Okay, so some of the main implications of this are that, for instance, the features that you see in the ventral stream, the tuning of the cells, would not reflect properties of shape, properties of images, but would reflect properties of transformations. So shape priors or natural images prior would be kind of irrelevant. Some of the other implications are, for instance, it requires homeostatic control of plasticity. It predicts tuning of various levels of the ventral stream. So let me go into this, and just to make sure I'm not too confusing, I'll speak alternatively about 2 different stages. One is development, the learning of transformations. And one is the mature stage where recognition happens. And recognition requires, if you want the learning, to have seen at least one example of the image, of the object you want to recognize, like a specific face, and then the recognition itself from another single images. So there are video and motion in development that's critical, but in the recognition stage, single images are enough, okay? So, start with the claim that the computational goal is to learn transformations, mainly, not only, but mainly affine transformation on the plane, and then become invariant to it. That there is this simple module, essentially simple cells, simple like cells that do essentially a dot product, a template matching with new images. And then it goes on saying that in order to make the memory requirements of this scheme feasible, there is a trick that evolution could have found of having a hierarchy of areas, layers. The first one essentially being invariant to small translation, and later one to the other affine transformations. And this greatly reduced the memory requirement of this schema, how many cells you need. And that this requires small apertures, small receptive fields in the first layer in order essentially to impose during development, if you want to train during development, the first layer, so in the ventral stream, V1, V2, only to translations. Okay, let me go through this 5 minutes. Okay, so the first is a theorem that has been quite influential in a lot of recent work with very large matrices, like at Google or Yahoo or so, which is this Johnson-Lindenstrauss lemma that essentially says that if you have a very high-dimensional object, like an image with zillions of pixels, you can take a relatively small number, still high-dimensional, but much smaller number of measurements, these projections, on the image, and this vector of measurements will still maintain all the similarity rankings that the original images have, okay? Now, this can be seen as saying that if you have enough measurements, it's not important which ones they are. Could be more or less random ones. For instance, in OCR, long ago, the way people took measurements on digits and letters in order to recognize it was to choose a more or less random set of lines. Once chosen is fixed. And then for each line, you count how many intersections it has with the character, okay? And so you get a vector of measurements. And this is a pretty good signature of each character to allow recognition. And there are many other similar measurements that you could take with similar results. So in our case, we'll just take any image, say, any frame of a video. Suppose a simple cell, this is a bit of a cartoon, but just to make it, suppose that you have simple cells during development, they see an object moving, translating, and they get imprinted with the frame 1, frame 2, and frame 3, and so on. And the measurements they will take later on will be just a template matching, a dot product, automatically done by the synapses w.x by each simple cell. And so you can think of each one of these rows in this matrix. Each one of these elements is a little matrix, is a frame of a video. You have time going from left to right. Each row is a little video. And it turns out that if you, under some assumptions, the following set of measurements gives you an invariant signature for the transformation you have looked. Suppose the tau 1, tau 1, 2, and so on are frames of something translating in X. Then suppose the cells record this during development. Then now in adulthood, what is done is that a new image is presented, F, and F is the template matching dot product, which each one of these templates, these frames, and then you take the max of this, for instance, or some other operation, and it turns out that this number that you get out for each row, so a vector, is invariant to translation. Okay. This is a property of, it's exactly invariant. This is a property of translation, rotation, scaling, all the elements of affine transformation. And so there is a receipt here to build an invariant signature. And furthermore, but the point is that you will need to store essentially a relatively large number of frames, because you will need to store all possible combinations of translation, rotation, and scaling. However, there is a property of the group of transformation, of the affine transformation, that you can be factorized as translation first, and then the rest, the scaling and the rotations. This is shown here. And this means that if I can do this, then I could have a layer doing first computing the signature, which is invariant to translation, and then the next layer or layers could compute a signature will become invariant to scaling and rotations. And it turns out that if you do it in this way, you have to store much less, like it's 10 to the 4 instead of 10 to the 8 or 9 elements. Here is just to show that, that one way to do this for evolution is to have to expose the first layer only to translations during development. And the way to do it is to have small receptive fields, because then locally, everything, every affine transformation is a translation. Oh, I see. OK. And so the idea is that if you have a hierarchical architecture with small receptive fields, small aperture, and then increasing apertures and going in this hierarchical way, you get you can stratify the different transformations, starting with translations to rotations, expansions, and transformations then that are not affine anymore, like, for instance, the one induced by rotation of a phase in depth or a change in expression of a phase. Now you can ask, what are the spectral properties of this example, of these matrices that I've shown before? It turns out that if you do this, these are simulation and doing PCA, you get, for translation, you get, this is translations in the vertical direction, you get Gabor-like principal components, quite similar to what you find in the physiology in V1. And then you, by the way, you get the same thing if instead of moving a natural image, you're moving a random noise. So these properties are independent of the content of the images, depend only on the transformations, in this case, the translation. So you get Gabor functions, and higher up you expect combination of spirals and combination of circle and radial patterns, circles for expansion and the radial patterns for rotations. And in fact, is what we find in terms of principal component higher up in adding a layer above V1. And this is work, ongoing work, so I don't have the whole story yet. But we expect to find things that are reminiscent of that cartoon down there. And the reason why we care about the spectrum is because if we assume that there is Hebbian plasticity at the level of these memory-based modules of these simple like cells, then slight modification of a Hebb rule necessary to avoid that it explodes, that it diverges, so homeostatic control of synaptic plasticity, result in changing the synaptic way in a way that they become the principal components that I showed before, or with some slightly different change, independent component of the spectrum. So, and in addition, the same memory modules, instead of exactly being invariant to affine transformation, can also learn semi-invariance to non-affine transformation. For instance, here, if you say it higher up in this Hmax model, you have a layer which, during development or during a plastic period, records how a face rotates in depth. Okay, so it stores these frames, and then just by doing these dot products and this simple operation on the output, you get much better invariance when you have to recognize a single image of a new face, of a different face, a different viewpoint. OK, so that's the story. There are a number of predictions here. One could try to interfere with development. The prediction would be that the natural world by itself does not matter. It could be noise. The important thing is to expose the developing system to a natural range of transformations. There is this prediction about class-specific modules, because these non-affine transformations like rotation of a face in depth can be learned and used only for a class of faces. The other affine transformation can be learned from any object and applied to any image. But for instance, if I want to be invariant to pose or recognize, estimate pose of a body, I need to have seen at least somebody change pose. So there are prediction of a few modules, like a place module for perspective, a face module for faces, body module higher up in anterior IT. We can discuss more if you want, but just let me show some of the students who have helped in this or previous work with HMAX. Thank you.